Hi, today's question is draw a bidirectional replication fork and label the origin of replication, the leading strands, lagging strands, and 5 and 3 ends of the strands shown in your diagram. You may stop video here, try to draw diagram on your own, and then compare your diagram with my drawing. So before I start explanation and drawings, I want to show some of the pictures that you usually can find in the textbook. This is a replication fork, for example, 3 and 5 prime ends, and here going to be 5 and 3 prime end. So this one new strand going to be 5 3, so this is going to be continuous strands that would be continuously built, and here we have 3 and 5, so this strand would grow in this direction from 5 to 3 direction, and as long as replication forks open and it moves in this direction, we would need more and more primers that would be here, for example, here and here, and they would emerge. Actually, they would be placed here with the help of the polymerase. This is enzyme, and these primers are made of RNA. Then they would be degraded and replaced with DNA, and so this would be discontinuous growing of this strand from the 5 to 3 direction, and probably this picture on the left is not correct, but it's just probably a little bit misleading, because it shows the only conception of the lagging strand and continuous strand. But on the other pictures that you also might find in the internet, for example, this is 5 prime end, and this is 3 prime end, 5 and 3. Sometimes you can find pictures on the internet that show something like this. New strand going to be going to go in the opposite direction, and shows like it's going to be continuous leading strand, and another one going to be 3 5, so this is going to grow something like this. So it's going to be lagging strand, but this is totally wrong. So let me delete these drawings first, and I will proceed with the rest of the explanation. So for example, if we take DNA strand, double strand of the DNA, and for example here in the middle going to be point of replication, and where replication bubble starts, and of course this is a linear presentation of the DNA, but in reality it looks something like this. So, and replication, point of replication going to be here, and for example, when bubbles start forming exactly at this location, so this is where the point of replication starts on both strands, and bubble going to grow in both directions and eventually form something like this. So it's going to move in this direction from the center and to this direction from the center. And let me draw a larger picture here of the replication bubble. So this is going to be 5' end and 3, 5 and 3, and this is going to be a region of replication where bubbles start to grow and new strand would be formed. I will draw a new strand with a new color. So for example, first premise would add primer here, and this is going to be 3 end and 5 end, and we know that new strand grows from the 5 to 3 direction, so it's going to be growing in this direction continuously. And what's going on the opposite side? Premise also add primer here, and it's going to be 5 and 3 end, and it's going to grow from 5 to 3 direction. So we see that on the both sides here we have continuous growing, so this is going to be both leading strands, this one and this one. And also what's going to happen on the other side from the replication point of replication, so this strand and this, how it's going to be formed. As long as we know that growth of the new strand only can be from the 5 to 3 direction, so new primers would be added as this bubble would grow in this replication fork would grow in this direction. So every primer would be look like this, and it's going to be growing in this direction. So but this is going to be discontinuous, and here also same picture. So 3' and 5', and this is going to be growing like this. From 5 to 3, and so we have here lagging strand and this one strand, and the opposite strand also will have lagging strands. So both new strands will have leading strand and lagging strand, and on the opposite new strand of DNA format we also can see lagging strand and leading strand. So this is a picture that probably would show you better conception how DNA replication works. Hopefully this was interesting and new information for you. Thank you for your attention today. Please subscribe to my new videos. If you like this video, thumbs up. and goodbye.